Hello, my name is Aaron Croke. I am a PhD student in NUI Galway under my supervisor, Professor Martin Leahy. I am presenting my poster on Nanostar modeling for uncovering some stem cell truths. My project is part of a European Union 2020 project called StarStem. The aim of this project is to provide an unprecedented understanding of the process of stem cell therapy and also provide doctors and researchers with a method of better measuring the healing process in stem cell therapy. There are a number of key questions when it comes to stem cells. Firstly, where do they go and how quickly do they get there? How many of the stem cells engraft themselves into the areas of interest? And how does the healing occur over time? The imaging technique that we are concerned with is photoacoustic imaging. Photoacoustic imaging is a non-invasive process using laser light to build images of tissue and other substances under the skin. Photoacoustic imaging takes advantage of the photothermal effect, heating the tissue, causing it to expand. This then produces a pressure wave, which is measured by the photoacoustic machine. Nanostars are used as contrast agents to increase the thermal response at the imaging site. In our project, we are uptaking these nanostars into stem cells, which are then inserted into the therapy site and imaged. The localized surface plasmon resonance is what determines a nanoparticle's optical absorption and response. While bulk materials' optical properties can be described in terms of their dielectric constant, nanomaterials' optical properties are dictated by the LSPR of the material. It is important to understand how this electron cloud interacts with incident light and other parameters, as the change in a couple of nanometers of the size can drastically affect properties of the material, such as the peak absorption. We can use optical modeling to model the absorption response of nanostars. Optical modeling is often beneficial because we can investigate new parameters that are difficult or time-consuming to test experimentally. We can then compare these different modeling techniques that we use to the experimental results that we receive to determine what modeling technique works best for our situation. The two modeling techniques that we will be discussing are FDTD and FEM. FDTD, or finite domain time domain modeling, is a modeling technique using a cube-shaped mesh to solve time-dependent Maxwell equations. FEM, or finite element method, is a modeling technique that uses a tetrahedral mesh to solve partial differential equations. In the nanoscale, small adjustments can have big changes. Small adjustments to geometrical parameters of nanomaterials, such as our nanostar, can have a large influence on the LSPR of that nanoparticle. On the right, we see our nanostar with adjustments on a number of geometrical parameters, and also how that affects the peak absorption in the near infrared region of that particle. In image A, we can see that we are adjusting the cone angle of our nanostar, and we can see how that affects the absorption peak of our nanoparticle, in particular in our near infrared region, which is our region of interest for our device. We can see here, as we increase the angle of the nanoparticle, we begin to shift the near infrared peak further and further into the near infrared region. In image C, we can see that we adjust the tip radius of our nanoparticle. We can see from this image that as we increase our tip radius, we drastically move our absorption peak into the near infrared region. And our last set of images, image E, we can see we are adjusting the branch length of our nanostar. And as we increase this branch length, we can see that it starts moving our infrared peak further and further into our infrared region, and it seems to stop having as a drastic effect around 40 nanometers in length. Another nanoparticle that we are concerned with are spions. Spions are super paramagnetic iron oxide nanoparticles. The additions of spions onto our nanostar particles means that we can use our nanostar particles not only for photoacoustic imaging, but hopefully also for MRI imaging, as MRI imaging needs a magnetic particle to be detected. Working towards 1064 nanometers. As I mentioned a slide or two ago, we are looking at moving the absorption peak in our near-infrared region to as close as 1064 as we can. This is because the commercially available ND YAG laser has an emission peak at 1064. If we can match our nanostar's absorption peak with the emission peak at 1064, it means that we'll have a much more efficient contrast agent when doing photoacoustic imaging. Below are two graphs. On the left, we have our FDTD modeling, and on the right, we have our FEM model. In blue, modeled on both graphs, is the bare nanostar, and in red is our nanostar conjugated with spions. We can see that the peaks are moving towards 1064. In our FEM model, we're showing a much higher peak in our near-infrared region than in our FDT model. But we can see that the addition of a spion onto our nanostar dampens the absorption peak in both cases. How does this compare to our experimental results of our nanostars? So model results can always be compared back to the experimental results that we can obtain. In our images above, on our left we see a bare nanostar in a high resolution TAM image. On the left we have a regular resolution TAM image of our nanostar conjugated with spions. It's important to compare and validate current models to make educated adjustments to our models as we move forward. The graph at the bottom of our slide here shows our absorption peaks experimentally measured in a UVIS spectrometer. In blue, we have our bare nanostar modeled, and in red, we have our nanostar with conjugated spions. We can see while the absorption peak is slightly over our 1064 region that we're looking for, we do see the same characteristic dampening with the addition of spions. So what's next? Next, we hope to move our modeling onto a photothermal model so we can evaluate the photothermal effects of the nanostars, both with conjugated spions and without. We need to do a photothermal stability study of our nanostars to see how they are affected by laser light and how stable they are. And then using these nanostars as contrast agents, we want to investigate other imaging techniques, such as NSOCT. Thank you for listening to my poster. I hope it was informative and I welcome any questions.